Okay, so hi everyone. I'm Reut Shalgi from the Technion in Israel. Here we are, the Faculty of Medicine, right on the beach in Haifa. I want to thank the organizers for giving me the opportunity to tell you about a fairly new and exciting phenomenon that we've been characterizing, which seems to be a hallmark of the transcriptional response to proteotoxic stress in mammalian cells. And this is transcriptional read-through. So transcription is highly regulated in response to stress. This is very well conserved in many organisms. This has been known for decades now, and I don't need to tell you too much about it. But actually, in recent years, it's been becoming more and more clear that there's a lot going on that's beyond just transcription regulation of genes. For example, splicing, which we've heard about a lot today, and it's actually also regulated in stress conditions. For example, we've shown a couple of years ago that it's widely inhibited in heat shock conditions in mammalian cells, but not completely, not globally, but rather in regulated way. Pol-to-pause release is highly regulated in stress. This is work from John Liss's lab, and this affects elongation rates. Polyadenylation is actually regulated in stress. This is work from John Manley's lab. And in general, it seems that there's many new emerging classes of non-coding RNAs that are being discovered, and many of them have unknown functions. And one such interesting class is read-through transcripts. So we've stumbled upon these transcripts when we've kind of stared at our RNA-seq data that was performed in a mouse 3T3 fibroblast before and after heat shock. Sorry, these labels skipped. This is the price you pay from switching from Mac to a PC. But in any case, it's pretty clear that, for example, for this gene, HNRNP-A2B1, which probably most of you have heard of, shows a fairly substantial read density beyond the gene end. And this stretches to distances about dozens of kbs in this case. And this happens only in heat shock and not in untreated cells. So if we wanted to look at it more systematically, we simply calculated the read density downstream to gene ends and normalized it to the read density inside the gene and call it a read-through score. And we see that in heat shock, compared to control cells, this read-through score is markedly upregulated. So at this point, we decided to team up with our collaborator, Anna Wilborg from Joan Steit's lab at Yale, who's been characterizing these read-through transcripts in osmotic stress in human cells. She called them DOGs. This is an acronym for downstream of gene transcript, and I'll use this throughout the talk. And what Anna found is that the DOGs are indeed continuous with their upstream genes. They remain mostly nuclear, and they have a lower frequency of polyadenylation site if you look downstream of the canonical one. But then together we decided to ask the following question. First of all, what is this phenomenon? How pervasive is this in different stress conditions, and which genes show this? The second question is, how is this regulated? Is this an adaptive stress response, or maybe simply a random failure? And third is, why is this happening? What are these read-through transcripts good for? So we decided to do a deep nuclear RNA-depleted RNA-seq, again in 3T3 mouse fibroblasts, and we exposed them to 3 different stress conditions: heat shock, osmotic stress, and oxidative stress. And Yuval in the lab devised a DOG discovery pipeline, which simply goes over the read density at 3' ends of genes and quantifies them and determines their ends, as you can see here. And we ran this pipeline on each and every condition separately and then compare the results. So for the sake of this presentation, I will always show RefSec gene tracks on top and these DOG tracks on the bottom. So we actually found that the phenomena is a lot more widespread than we thought. Almost 5,000 genes had a DOG in at least one stress conditions, and 1,500 DOGs were common to all stress conditions. Now we also find these little tails in unstressed cells, as you can see here for HNR and PA2B1. We found them pretty often, not that often, but but we find quite a few of them. And as you've heard before, these may represent natural termination sites because, of course, the 3' end of the gene is not a natural termination, but rather the cleavage and polyadenylation site. However, when we compare the length of these transcripts, we see that they're getting almost universally much, much longer. This is the distribution of lengths here. And here we compare dog by dog in untreated cells compared to the 3 stress conditions. And you can see that this picture is actually very well representing what's going on. They become much, much longer. We also looked at their expression levels, again, by quantifying the read-through score in all of the conditions. And here I'm showing you a distribution of the fold changes in the read-through score in the different stress conditions. And you can see that it is markedly shifted towards upregulation. So now to our next question, which is, how are they regulated? Is it an adaptive stress response or simply a random failure? So one simple explanation could be that the cleavage and polyadenylation machinery actually works less efficiently in stress conditions, and then perhaps just a failure to cleave and polyadenylate leads to elongation, transcription elongation that simply continues and fails to terminate, and this just happens more frequently in stress conditions. However, if this were the case, then we would expect that the more highly expressed the gene is, the more high the read-through would be. And the more highly induced the gene is, the more highly induced the read-through would be. And this is not what we observe. We see here a fairly low correlation between the gene levels and the dog levels, as well as between the gene fold changes and the dog fold changes in the different conditions. So we conclude from that stress-induced transcriptional read-through is likely not due to random failure, but is rather some kind of an adaptive response. We wanted to look at Pol2 occupancy that goes along with read-through, and for that we took advantage of a ProSeq data from John Liss's lab before and after heat shock. And we looked at the ProSeq occupancy in different windows downstream of gene ends in dog positions. And indeed, we saw that there is a much higher enrichment of Pol2 occupancy in dog regions in heat shock compared to control. And the difference is becoming more and more larger and larger the more downstream we go into downstream of the gene. The next thing we wanted to do is to compare genomic features, such as sequence features. But first, we had to come up with the appropriate control. So we want to look at pan-stress dogs. These are the 1,500 dogs that are common to all stress conditions. And as control, we defined a set of non-dogs. These are genes with no evidence of read-through in any of the stresses that we have. And they are matched with expression levels to the pan-stress dog genes. This is very important. And if we perform the motif analysis this way, we see again that the AAU-AAA canonical polyadenylation signal is significantly depleted in dogs versus non-dog genes, but also we find some other motifs, including AC and GU-rich sequences that are highly depleted in dogs compared to non-dogs. So we see different sequence signatures for these read-through transcripts. And now to the last part, or the last question, which is, why is there a transcriptional read-through? What are they doing? And the short answer is, we don't know yet, but we do have some clues. And the first clue came when we stared at quite a few of these, and oftentimes we see here a gene close to the very end of these dogs. So we decided to quantify this more systematically and look at the dog frequency with relationship to the distance to the nearest 3' neighbor. And as you can see here, dogs really like to be in the vicinity of other genes. So this actually has a fairly immediate implication because dogs tend to be in the vicinity of neighboring genes and they get longer in stress. This has implications on potential antisense transcription with opposite strand expressed neighbors. And indeed, when we count the number of genes that are potentially affected by these longer dogs, we see that this number goes up significantly, especially in osmotic and oxidative stress. Nevertheless, this trend of dogs liking to be in the vicinity of other genes holds true whether the neighbor is on the same or opposite strand, and also whether the neighbor is expressed or not. So antisense cannot be the sole reason for dogs to exist. There has to be at least another additional explanation. So this prompted us to think about a potential relationship to chromatin features. So for that, we took advantage of some mouse ENCODE data of histone marks, and we looked specifically at 3 regions: promoter regions, ends of genes, and downstream regions of dogs versus non-dog genes. And here's the data. So for this chromatin mark, H3K4-methyl-3, this is a well-known promoter mark. We see no difference between dogs and non-dog genes. However, these 2 regulatory chromatin marks show a significant enrichment in dogs compared to non-dog regions in the dog regions. So, and we saw the same phenomenon for some elongation marks. So is it possible that dogs tend to be in regions of higher chromatin accessibility? So to look at chromatin accessibility, we downloaded DNA-seq data as well as ATAC-seq data. And again, we see in the dog regions a significantly higher enrichment in these open chromatin marks in dogs compared to non-dogs. So just to quickly summarize what I've shown you today, there is a vast induction of transcriptional read-through in many proteotoxic stress conditions that generate thousands of long nuclear transcripts up to dozens of kbs beyond gene ends. These are likely not due to random failure, but rather some kind of an adaptive response. They have distinct sequence characteristics, and we see Pol2 in them. And as to what are they doing, we still don't know, but we have a few hints related to antisense transcription and perhaps a role in maintenance of open chromatin. And finally, I want to thank everyone that were involved. First of all, Anna Wilborg from Jones Stites Lab at Yale, and Niv Sabat and Yuval Wiesel from the lab, who did all the bioinformatics analysis. And thank you for your attention.
Thank you. Okay. Any questions for Reut?
So very nice. I was confused about the why and the how, whether they're matched in a way. So you said, look, this is probably regulated because it doesn't correlate very well with expression. And then at the end, you showed it has to do with chromatin marks. And so one could argue that what has happened is it's not related to expression level, it's related to chromatin marks, and just sort of that's it. And so that's the component. So the proof for regulation that it's not an artifact, you could argue it happens when those chromatin marks are there. These are the guys that are affected. It's just what happens under stress. These are more prone to so you know, if you, for example, if you could show that these guys that are affected have specific function that they want to, you know, say, put, you know, reduce or something like that.
True. So first of all, just to mention, all of these are expression matched, so there's no confounding factor for expression. However, indeed, before the stress, these chromatin marks are not in stress conditions. Before the stress, the region is more open. But still, the region is more open, yet we see differential induction of these dogs in different stress conditions. And we cannot simply explain that by this data, but it's true that this region is more permissive for their expression. Why do they continue so long? Why is it selective in some genes and not others? That's still open.
Okay, so we have one last question.
Hey, so we know in plants, well, I say no, but we suspect in plants where we see a lot of these read-throughs, particularly in, we see a lot of the read-throughs and they change significantly in degradation mutants. So if you disrupt the XRN complexes, for example, then you see an increase in read-throughs. So is there any evidence that you know of that perhaps what you're seeing here under stress conditions is actually not necessarily related to expression or antisense, but is that the stress condition is actually disrupting or changing the degradation program, and that's what's causing your increase in read-throughs.
That's, well, I can't see you here behind this poll, but I'll answer anyway. So that's it's something we considered, and it's quite possible. However, there's two pieces of evidence that would argue against this. First of all, these are not simply the RNAs that were remaining after cleavage and polyadenylation and just failed to terminate, to be degraded. These are in fact continuous, and we tested that. The second thing is that we knocked down XRN2, and we did not see an effect on read-through. It doesn't mean that there are several XRNs that might be acting in concert, and we didn't test for that. And the third point is that we do see more polymerases in heat shock. This is here. Compared to control. So they do go the longer distance, and they do not go to that distance in the control condition. So this is not simply a failure to degrade this, where this is degraded very efficiently in unstressed cells and not in heat shock, for example.
That's exactly what you would expect if you disrupt the XRNs, because, well, there's this idea anyway that there's a torpedo model. So the XRNs latch on to the transcript as the polymerase keeps going. And if it reads through the termination site, the polymerase just keeps going, but it gets chased down and bumped up by the XRNs. So if you disrupt the XRNs, you'll see exactly what you see there. The polymerases go further.
We did not, yeah.
Okay, maybe we should take this conversation for the coffee break.
Definitely.
Yeah? All right. Thank you. So let's thank Reut again, please.